We will now look at the Calvin cycle. Remember that ATP and NADPA are made in the light reactions so that they can go to the Calvin cycle, which is the process of fixing carbon into sugars. All right, so let's look at Calvin cycle. Also, I'd like for you, and I'm hoping that you're reading after you're watching these videos, or maybe before watching these videos, screencasts that you're reading out of the textbook. Light stimulates the Calvin cycle. Calvin cycle is called light-independent reactions. It's really, I wish they would call it the not directly dependent on light reaction, because Calvin cycle does depend on light. If there's no light, guess what? There's no ATP and no NADPA. Light is required to make the ATP and the NADPA. So when there's no light, photosynthesis just stops. It's not like the Calvin cycle can keep running without light. The Calvin cycle has to have ATP. Look right there. ATP. ATP going into the Calvin cycle. NADPA going into the Calvin cycle. So if there's no light, there's no ATP or NADPH made, therefore no Calvin cycle. So, Calvin cycle uses NADPA and ATP, which are generated using energy from light. Two other processes connect light reaction with this carbon dioxide fixation pathway. Both connections are indirect but significant. Light-induced pH changes in the stroma activate some Calvin cycle enzymes. Remember, the protons are pumped from the stroma into the thylakoid lumen. Remember when we looked at the electron transport chain? The protons in the stroma, the light green part of our model of our chloroplast, protons are pumped actively into the interior of the thylakoids. Well, this area inside the thylakoids has an abundance of hydrogen ions, so it's more positively charged, and in the stroma would be more negatively charged. So, and also more, if you have hydrogen ions, it's going to be more acidic, so there will be more a lower pH in the thylakoid, a higher pH in the stroma. Well, that change in pH in the stroma will activate some Calvin cycle enzymes. Changes the pH in the stroma from 7 to 8. That's a tenfold decrease in hydrogen ion concentration. This favors the action of Rubisco, which is an enzyme that is required to get the Calvin cycle started. Okay, another connection of the light reaction with the Calvin cycle is light-induced electron transport reduces disulfide bonds in 4 of the Calvin cycle enzymes, activating those Calvin cycle enzymes. I wouldn't worry about all the details on that, but you just know electron transport will activate some of the Calvin cycle enzymes. Okay, so let's look at the Calvin cycle. You can see that the Calvin cycle takes place in the stroma of the chloroplast. There are those dark green pigment-filled, chlorophyll-filled thylakoids. So the Calvin cycle is taking place in the stroma. This is where the incoming carbon dioxide lands. So carbon dioxide coming through, diffusing through the stroma, the holes on a plant leaf, in through the spongy mesophyll. We'll be looking at the structure interior of a leaf again, and will enter plant cells and will enter the chloroplast. And here comes the carbon dioxide into the stroma. Carbon dioxide combines with a substance called RUBP. It also combines with an enzyme called Rubisco. Rubisco is the most plentiful enzyme in the world. It's found in all plants and photoautotrophs. So even though the enzyme Rubisco is not written here, you know that carbon dioxide combines with the enzyme Rubisco, substance called RUBP. CO2 combines with its acceptor, RUBP, the enzyme Rubisco, facilitates the formation of a substance called 3PG. So we've got carbon being fixed or transferred into a different substance called 3PG. This part of the Calvin cycle is called carbon fixation. So it's carbon dioxide plus RUBP with the enzyme Rubisco forms 3PG. Then 3PG is reduced to G3P. Oh, reduction. Oh, getting a hydrogen atom. Remember, being reduced, you're getting a hydrogen atom, therefore you're getting the electron. So this part of the Calvin cycle is called reduction and sugar production. 3PG is reduced to G3P in a two-step reaction that requires ATP and NADPH. Oh, this is where the ATP and NADPH come in. Where were they formed? In the light reactions. So the light reactions make the ATP and the NADPH that are required to reduce 3PG to G3P. What about the sugar production? 1/6, center that a little bit better, 1/6 of the G3P is used to make sugars, C6H12O6. That's the purpose of the Calvin cycle, make sugars. But only 1/6 of the G3P is used to make sugars. So if a plant is not photosynthesizing very much, it's not going to put out a whole lot of sugars. So what about the rest of the G3P? The remaining 5/6 of the G3P are processed in the series of reactions that produce rump. In other words, most of the G3P is converted into rump. Then rump is converted into RUBP in a reaction that requires ATP. So here's some of that ATP that was made in the light reactions. Some of that ATP is used to convert rump into RUBP. Why do you need RUBP? Oh, this is a Calvin cycle. RUBP starts the cycle all over again. Carbon dioxide plus RUBP with the enzyme Rubisco makes 3PG. So the reason this is the Calvin cycle is because RUBP has to be regenerated to be present to combine with carbon dioxide. So that's the Calvin cycle, just a brief description summary of the Calvin cycle. Let's look at it again. Carbon dioxide coming into the chloroplast, into the stroma, combines with RUBP. There's the enzyme Rubisco, facilitates the reaction to make 3PG. This stage is called carbon fixation. 3PG is a 3-carbon sugar. 3PG is reduced to G3P. Remember, reduced? Oh, you're picking up hydrogen atoms. So 3PG being reduced to G3P requires the ATP and NADPH from the light reactions. You don't have light, you don't have ADP and NADPH. A little bit of the G3P, about 1/6 of the G3P is used to make sugars. Yay! That's the purpose of the Calvin cycle. The remaining 5/6, oh, and let's go back. Reduction in sugar production is converting of 3PG into G3P, some of that G3P used to make sugars. Then most of the G3P is used to make rump, RUMP. The rump is converted with the help of ATP by chemical reactions into RUBP. And remember, you've got to have RUBP to start the cycle all over again. So that is the Calvin cycle. 6 turns of the cycle gives you 1 molecule of glucose. So that's the Calvin cycle. I would encourage you, if you've not read through section 3, so you need to have read, and if you haven't done it, do it now after watching these screencasts. Hopefully me just talking through it simple version will help you understand it a little bit better as you read it. But you need to make sure you have read sections 10.1, 10.2, 10.3, because the next topic is going to be photorespiration, C3 plants, C4 plants, which is something that we did not cover in 10th grade. So need you to nail down 10.1 through 10.3, have a good understanding of it.